Welcome to the PMN tutorial video series. Today, we are going to teach you how to use the Enzyme Activity Curation form of the Plant Metabolic Network. This form allows you to curate new enzyme activities into PMN. The curation process has three main steps. The first step is to enter your contact information. After you do this, you will provide your database details. Lastly, you will be asked to add your enzymes, reactions, and enzyme activities. Before you begin this process, you will need to confirm that the enzyme activity belongs to one of the 582 species currently in PMN. You have a gene or protein ID that matches the annotation used for that species, and that the reaction already exists in either plant psych or meta psych. Alright, now you are ready to begin. To start the process, go to pmn.plantpsych.org/enzyme curation. On this page, please note that there is an FAQ section available on the page and there is a curation summary panel on the right. The curation summary will keep track of all of the information that you provide and will also allow you to edit as you go. You can access these at any time during the process. You will begin by entering your name, institution, and email address into the blue Curator information box. Please note that only your name and institution will be publicly displayed. Your email address is used only if we need to contact you. Once you've done this, click the Next button, and your information will appear in the Curation Summary panel on the right side. Next, enter the database you are curating into the pink database selection box next to where it says Psych Name. You can include or omit psych at the end of the database name and it will be accepted. For this tutorial, we are going to search in AeroPsych. Then click Next. Now you can add your enzymes. First, you will select your accession information. At this time, we only accept identifying enzymes by an accession that matches the ones already in the database, so that will be the only option that you can select. We hope to offer other means of identifying enzymes in the future. Now we are going to enter the enzyme ID. This enzyme doesn't have to be in our database, but it does need to have an accession in the same format as the ones that are. Therefore, you will need to provide the protein accession number and add a common name if available. If the format is recognized, examples will appear automatically. Then you will click Add Enzyme, and it will add to your creation summary panel. Repeat this step for all enzymes. When you have finished entering all of your enzymes, click Done Entering Enzymes. The next step is to add reactions using their reaction IDs. If you don't have a reaction ID, open another window without closing your current window. There is no autosave, so all data will be lost if you leave the page unless you download it before doing so. Go to pmn.plantpsych.org, select the plant psych database or the database you're curating in. Search by compound name or reaction name. You are looking for the biopsych ID. This ID is typically all uppercase and either starts or ends with RXN. You can find it here on the reaction page. You can also find this ID in the URL. Once you have a reaction ID, select Yes, I have a reaction ID. Please note, only reactions from plant psych or meta psych are accepted, and invalid IDs will not appear. Then click Add Reaction, and confirm it appears in the summary panel. Repeat as needed, and when finished, click Done Entering Reactions. Now we are on our final step, adding new enzyme activity. Each enzyme activity represents a statement. This enzyme catalyzes this reaction. For each activity, you will assign a reaction to each enzyme, add a citation, a PubMed ID is preferred but a DOI is accepted, and then choose the type of experimental evidence. Please note we do not accept computational evidence or gross phenotype evidence, only evidence that specifically identifies the reaction being catalyzed by the enzyme. Regarding the evidence types, the first three options are for native hosts, and the next three are for heterologous systems. As for the purification levels, you have the choice between purified, enzyme isolated and tested in vitro, partially purified, mixed protein extract tested, or unpurified, in vivo evidence. After choosing the experiment evidence, click Add Enzyme Activity to save each entry. Keep in mind, one enzyme can have multiple reactions. One reaction can have multiple enzymes, and multiple pieces of evidence for the same activity can be indicated by creating separate entries. Once you have finished, click Download Results and email the file to curator@plantpsych.org. At any time, you can go back and modify entries by using the Edit button next to each item, and you can view all of your entries in the summary panel. Since there is no autosave option, if you need to come back to your entry at a later time, click Download Results when you've reached a stopping point. Then, when you come back to the curation form, on the first page, you will click Continue from Previous Curation and upload your downloaded file. We hope you found this video tutorial helpful. If you have any questions, please reach out to us via the feedback form on our website at plantpsych.org. Stay tuned for more tutorial videos on how to use the Plant Metabolic Network funded by the National Science Foundation.